rising. So looking forward to that. The duration of this webinar is about an hour, and we will see what time we have for questions and answers at the end. But in the meantime, welcome to you all. I'm just going to introduce you to Dr. James Clement. He's our medical advisor for motor neurone disease. He's a general neurologist with subspecialty expertise in neuromuscular disorders and electrodiagnostic medicine. Principal research interests are motor neurone disease and neuronal intranuclear inclusion disease, or NNIID as it's called. He's employed as a neurologist at Tauranga Hospital and is also in private neurological practice in Cambridge. In addition to his clinical and research roles, James serves as president of the Neurological Association of New Zealand, is a member of Pharmac's Rare Disorders Subcommittee, serves in the advisory board of Punaha Eo Neurogenetics Registry of New Zealand, and is the medical advisor to us as MND NZ. So welcome, James, and thanks for your connection to Dr. Bedlack from the past, and I'll hand over to you to introduce Dr. Bedlack to us all.
Thanks, Mark. Kia ora koutou, everyone. And as Mark says, fantastic to have such a great turnout for what I think is going to be an excellent session. So it's my great pleasure to welcome Dr. Richard Bedlack, MD, PhD. Dr. Bedlack is the Stuart Hughes and Went Distinguished Professor of ALS and the Director of the ALS Multidisciplinary Clinic at Duke University in North Carolina. I've only met with Rick once in person back in 2003. Both of us remember that. At the time, he impressed me not only with his passion for motor neurone disease, but also with his incredible dress sense. I've followed his career ever since, and over the course of that career, Rick has published more than 160 scientific papers on motor neurone disease, has been at the forefront of clinical trials and therapeutic development. But equally impressive are the numerous honours recognising his contributions to ALS research, patient care and medical education, for which he's won the Forbes Norris Award and the American Academy of Neurology Patient Advocate Award, among many other awards. Throughout this time, I've also admired his outstanding contributions, well, not only his outstanding contributions to ALS research, but also his hope and optimism during a period where ALS research has been often slow and challenging. And I have enormous respect for his commitment to bringing hope to patients and families through the careful scientific evaluation of off-label treatments, such as an ALS Untangled, which is a website that Rick started up, and also through his pioneering work in studying people who have experienced unexpected ALS reversals. So in summary, I would say Rick is one of those rare physicians who combines world-leading expertise, but also a genuine compassion and also passion for improving the lives of people affected by motor neurone disease. It's a real privilege to have him with us today. Please join me in welcoming Dr. Richard Bedlack.
Wow, my goodness, James, that's a beautiful introduction. I'm over here blushing. Thank you so much for having me. I'm going to talk tonight on motor neuron disease perspectives, progress, and positivity. And just to clear up a couple things right at the start, you will hear me use the term motor neuron disease and the term ALS interchangeably. Over here in the US, we usually call this ALS, but I know it's not called that everywhere. And you also may see me spell neuron differently. I noticed that you add an E at the end. We don't usually do that. But either way, I hope we can agree that the motor neurons look really good embroidered down the side of a pair of pants. That's actually a real pair of my pants in that picture. So I want to just start off by giving you some disclosures. I do have some financial support from my research from a company called Medicinova and from the Healy Center. I have financial support from my clinic, from the ALS United chapter of North Carolina, and I've got consulting support from several different companies. What I do for these companies mainly is either help them to design more patient-centric clinical trials or serve on their data safety monitoring boards and help them watch for the safety of people participating in their trials. So I've got a lot of things I want to try to cover. We'll just spend a short time on each one of these, but I was asked to present some of my favorite recent clinical and research developments. And so under clinical, I'll talk a little bit about ALS subtypes. I'll talk a little bit about our first home run drug for one of those subtypes, which is called tofersen. I'll talk about some new data on an old drug called riluzole, as well as an old drug called dextromethorphan and quinidine, also known as Nuedexta. I'll tell you about some exercises, specifically respiratory strength training exercises that we believe are very promising for slowing the progression of the disease on certain measures of breathing and cough. I'll tell you about a specific diet that looks like it's associated with the slowest progression. And I'll tell you about something very surprising that's come about in the past year or so related to these drugs called GLP-1 modulating drugs. And I'm sure most of you have heard about these drugs. They've just exploded. They're mainly used for diabetes and weight loss, but I've heard of people now using them for lots of different things, and they might not be a good thing for people with ALS to take. I'll show you some data on that. And then when we get to my favorite research updates, I'll tell you about some attempts to target other causes of ALS, and I'll tell you about my work on understanding outliers. And then finally, at the end, I'll tell you about some things that are happening around the world. I call it something for everyone. I think hopefully there's going to be something in there that everybody can take away from this talk and go and talk to their own doctors about. So let's start with the clinical developments. So for a long time, we've recognized that ALS looks different in different people. And this is a really nice graph that kind of illustrates that. So we've got what we call a spinal or limb onset form of ALS, where the weakness starts in one arm or one leg. We've got a bulbar onset form where it starts in the speech and swallowing muscles. We've got primarily lower motor neuron forms of ALS that lean towards something called progressive muscular atrophy. And we've got primarily upper motor neuron forms of ALS that we call primary lateral sclerosis. We've got different patterns of weakness in ALS. We've got one that's called the pseudo-polyneuritic form of ALS, which starts in a stocking and glove pattern. We've got a form of ALS, believe it or not, that stays localized to one side of the body, at least for many years. It's called the hemiplegic or Mills variant of ALS. We have something called the flail arm variant, and we have something called the flail leg variant. And those are primarily lower motor neuron forms of ALS that stay localized to the arms or the legs for a very long period of time. Different. We've also known for many years that the progression of people with ALS is very different. And primarily, we used to measure progression based upon survival, but there's so many artificial things that we do now to prolong survival in people with ALS. It's not a clean measure of progression. So one that has been used now for more than a decade is called the ALS Functional Rating Scale, the rate of decline in the ALS Functional Rating Scale. So in case you don't know what this is, it's 12 questions that we ask patients about different functions like their speech and their swallowing, their ability to cut up food, their ability to walk. And each one of these questions is scored between 4, which means that function is perfectly normal, and 0, which means it's not there at all. And so it's been said that on average, people across the world with ALS lose about 1 point per month on this scale. But these are some individual traces of single patients whose ALS FRSR score was followed for many months. And you can see how different the slopes are. I mean, there's that one patient there, this sort of a blue line that goes the furthest off to the right of the screen. I mean, they're hardly changing, and they're all the way out to, like day 70 or month 78, hardly changing. And then there's some people in red who really drop fast. And so we know it's a wildly different disease in terms of the progression of functional loss. And so people used to argue, does that mean it's a different disease? Or is it just that there might be modifiers that determine what the same disease might look like or how fast it progresses? And in recent years, I think the question has been answered pretty thoroughly. It is not the same disease in different people. And I'll present 2 really strong arguments for that. The first is this study from 2019. Here they took samples of brain tissue from 148 people with ALS, and they used 2 techniques called RNA-seq and immunohistochemistry to characterize what they saw happening in the cells from these brain samples. And interestingly, it wasn't the same. So there were three sort of distinct groups in terms of what they saw happening in the cells. They called one the ALS glia group, which was characterized by activated microglia. That could also be called a neuroinflammation group. They had an ALS ox group, where there were lots of markers of oxidative stress and stress to something called the endoplasmic reticulum. And then there was this group in the middle that they called the retrotransposon group. So retrotransposons are pretty fascinating and pretty terrifying. They're tiny little fragments of DNA that can jump around and insert themselves into parts of our normal genes, and they can really disrupt what happens in cells. And so they act a lot like retroviruses, like the HIV or the HERV-K retrovirus. So three distinct molecular pathologies in people with ALS. And I hope that this is even more convincing. It's very clear that there are many different causes of ALS. And we divide those causes into two categories, genetic and sporadic. So genetic ALS means we can find a single broken gene that we think is causing the disease. And there are now more than 40 different genes, any one of which can be broken to be able to cause this disease. So the vast majority of people don't have one of those. And we call that sporadic ALS. So 85% of people, we can't find a single genetic mutation that we think is the cause. And it's starting to look like there are probably combinations of susceptibility genes that open the door, and then exposures that allow the disease to come in. And some of the exposures that have some evidence are military veterans. Turns out veterans are about twice as likely to get ALS compared to non-veterans, no matter when or where they served. Elite athletes, especially athletes that participate in contact sports, like soccer players and NFL football players, are more likely to get ALS. And then there seem to be clusters of ALS around bodies of water in the world where there's lots of blue-green algae. And we have one of those clusters in the state of North Carolina. So this idea that ALS is more than one disease, it's important for more than just academic reasons. You know, we've been trying for 100 years to find one home run drug for everybody with ALS. And I don't think that's going to happen. I remember growing up, we used to talk about finding a cure for cancer. And now, of course, we look back and we laugh. Of course, there wasn't going to be one cure for all cancer, because cancer is a very different disease. It requires precision therapy based upon the types of cells and how big the tumor is and how many different places it is. People get very different treatment regimens. And so now we've used this understanding of ALS subtypes to develop what I think is the first home run drug in the history of ALS. It's called tofersen, or in the United States, brand name Qalsody. This drug belongs to a class called antisense oligonucleotides, and it is specifically designed to treat people with one genetic form of ALS, the kind caused by mutations in the SOD1 gene. So what this drug does is it prevents that bad gene from making a bad SOD1 protein. So the development of this drug was very interesting for a number of reasons. This is not a very common form of ALS. There's probably 1% to 2% of everybody with ALS in the world living with this form. So trials are very difficult to do. Kudos to the investigators who got 2 clinical trials done. The first one showing safety and target engagement, and the second one, which was really sort of the pivotal trial to show effectiveness. At first, we thought it was a disappointment, because at the end of 6 months, it was not clear that people who got tofersen were doing much better clinically compared to people who got placebo. But they did something really important. They added a biomarker, something called neurofilament light chain. This is a chemical that we think leaks out of any kind of damaged nerve cell, so it is not specific to ALS. However, it is markedly elevated in most people with ALS. And in this trial, after just a few months, people on tofersen had a more than 50% reduction in neurofilament light chain, whereas people on a placebo didn't really have any change. And so the FDA said, this looks really promising. Like, it seems like you've dramatically decreased the loss of motor neurons based on this biomarker, keep going, keep following these patients. And sure enough, as we follow people out, it became very clear that people who started this drug right away were doing remarkably better than people who were randomized to a placebo. And in fact, now that we continue to follow people, we can see that more than half of the people who get this drug and stay on it for 2 years have stopped progressing completely. And about 25% of people are getting much stronger. So that's why I say this is the first home run. So I mentioned that we've been trying to develop one drug for everybody for 100 years without much success. I would say that we've had one modest success, and that is the drug riluzole. So this is an oral medication. It now comes in multiple different forms, tablets, suspensions, oral films. We think it probably has multiple mechanisms of action. Primarily, we think it reduces excitotoxicity by inhibiting the release of something called glutamate, but it also seems to reduce neuronal hyperexcitability by blocking sodium currents. And believe it or not, this was FDA approved now 30 years ago, based upon 2 really large, randomized, long duration, placebo-controlled trials. And those trials showed clearly that people who took this at 50 milligrams every 12 hours lived longer without being attached to a ventilator compared to people on a placebo. And so it's been around a long time. I'm always amazed when I see that the use of it is not very high, because it seems to be pretty safe and it seems to be well tolerated. And it's really the one thing that has this kind of data behind it for everybody. And I think part of the reason it's not more widely used is because of the effect size. So people have looked back over these trials and they've said, wow, the difference in survival between all those patients who are on the drug and all those patients on the placebo, it's really only about 2 to 3 months. And so the way this is often presented in clinic is, well, we do have this drug, it's only prolonged your life by a couple of months, and there's a chance it could hurt your liver. Well, it's really difficult to extrapolate the results of a trial to an individual person sitting in front of you. And I'll give you an example of why that's kind of silly. I'm on one medicine. It's a blood pressure medicine. When my doctor decided what to put me on, she did not come in and say, Richard, I want you to take this medicine. It will lower your systolic blood pressure by exactly 13 millimeters of mercury. No, of course she's not going to say that, even though that might be the difference between drug-treated and placebo-treated patients in a trial. That doesn't mean that's what's going to happen to me. I might get a huge effect and pass out, or I might get no effect at all. And thankfully, people have studied riluzole now in what we call real-world studies, where they've compared large numbers of folks who are taking it to a matched sample of patients with ALS who are not taking it. And in fact, in the real world, the survival benefit looks like it could be much larger, possibly as much as 19 months. So in my opinion, most people who want to try to slow the progression of the disease and prolong survival should consider this medication. So we just recently learned something about another older medication, and that's this one called dextromethorphan and quinidine. Goes by the brand name of Nuedexta here in the United States. This is another oral medication. It's something called a sigma-1 receptor agonist, and it was FDA approved for a specific symptom that some people with ALS get called pseudobulbar affect. This is where they lose the ability to control their laughing and crying. And again, this was approved a long time ago, 16 years ago now, and the trials were very good, multiple randomized, double-blind, placebo-controlled trials showing not only reduction in frequency, but reduction in severity of pseudobulbar affect episodes. But what was interesting in the years since those trials is that some of us who were prescribing it noticed other things happening to our patients. For example, we noticed that some patients' speech seemed to dramatically improve. Some people's swallowing seemed to dramatically improve. And so in the past few years, there have been some trials of this medication specifically targeting bulbar function. So some of these trials actually enrolled people without any pseudobulbar affect. And these were also good trials, randomized, double-blind, and placebo-controlled. And they sure enough showed that it can improve speech, swallowing, and even the ability to handle oral secretions. Now, one of the challenges of this exciting data is that no one has taken this medicine forward to any regulatory agency and tried to get it approved for the treatment of bulbar dysfunction. So in the United States, if I want to try to improve someone's speech with this medicine, I can't prescribe it. Insurance won't pay for it. A person can buy it themselves, but it's very expensive. So the only way I can prescribe this and get insurance to pay is if I can show that there's pseudobulbar affect in my patients that's disturbing their quality of life. So hopefully that will change. So let's turn our attention away from medications for a second to another topic that I often get asked about, and that's exercise. And this is interesting how this has evolved. So believe it or not, I've been in the ALS field for 25 years now. And I remember when I first started, the person who taught me saying, whatever you do, don't let your patients with ALS exercise. They'll burn out their motor neurons faster. I have no idea why they told me that, probably because their teacher told them that, but I can't find any study that suggests that that's true. In fact, there have now been studies suggesting that exercise is probably a good thing for people with ALS, that people with ALS who exercise generally feel better and probably live longer compared to people who don't. Now, for the most part, we don't know exactly what the right type of exercise is for people with ALS. We need more studies. And in fact, it might turn out that there isn't one universal type of exercise that is the right thing for everybody. But there is this new form of exercise that looks like it might help most people. It's called respiratory strength training. And this is basically a breathing exercise. So you use these valves and you either blow into them or you suck into them. So you're working against the resistance with your inhalation or your exhalation. And there have now been two really well-designed, randomized, double-blind, sham-controlled trials where they showed improvements. So one of the trials showed improvements in people's ability to breathe out, what's called the maximum expiratory pressure. And the other one showed not only that, but also an improvement in cough strength. Peak cough flow improved. And that second trial finally showed a slowing in the rate of decline in bulbar dysfunction by more than 50%. So in my clinic now, my speech therapist teaches these to all our new patients. Another question that I've been asked a million times in the past 25 years is, what about diet? Well, we've known for many years now that weight loss is bad for people with ALS. When people with ALS start to lose weight, they often start to progress faster. And it's not easy for people with ALS to maintain their weight. Sometimes they have problems swallowing. Sometimes they have trouble getting up and getting food to their mouths. So they have to ask people, which I know gets frustrating. There's evidence that people with ALS are actually hypermetabolic, that they might burn more calories. So they might need even more calories than they did before they got ALS. And so for a long time, what we told people is just calories, 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 calories. But we didn't have anything more specific. And then last year, the folks at Columbia published this study. This is not a randomized double-blind placebo-controlled trial. It turns out those are very difficult to do when it comes to diets. Part of the reason it's difficult to do is because human beings don't want to stay on the same diet for 6 months or 12 months. But this is an association study where they followed a large group of people and they asked them questions about what they were eating. And it turns out there was a surprise. The diet that was associated with the slowest progression was one called a high glycemic index or high glycemic load diet. So glycemic index is a measure of the blood glucose raising potential of a food compared to a reference food. So the glycemic load is obtained by multiplying the glycemic index by the amount of carbohydrates in a serving of that food. And to simplify all that, I've put that diagram there from the Cleveland Clinic. These are some different food groups that most of us know. And you can see that the ones with the highest glycemic index are things with a lot of sugar or a lot of carbohydrates. These are the exact types of diets that most of us are told to avoid for other problems like high blood pressure or obesity or diabetes. But it turns out that for right now seems to be the diet that's best for slowing progression. And then last but not least, I promised I would mention this little surprise. So this first came to my attention in a case report that was published showing a person with slowly progressive ALS who was started on Ozempic for diabetes and had their progression just accelerate. And there now is also a case series published in Neurology in 2025, suggesting that this entire class of medicines, the GLP-1 agonists, but also the so-called DPP-4 inhibitors, using those is associated with much faster progression compared to people who don't use those or use other treatments for their diabetes. And I have to say, I saw this in one of my own patients. Again, I had a patient that was doing really well. All of a sudden, their progression seemed to have exploded, and I was trying to figure out why, and I didn't see anything new on their med list. So I was questioning them in every way. What have you changed? What have you changed in your diet, your supplements? And they said, oh, I forgot to tell the nurse. My primary care doctor started me on Ozempic for my diabetes, and that probably was what it was. All right, so those are some of my favorite clinical updates. Let me tell you now about some of my favorite active research. So I told you about tofersen, and I think that's where the field needs to go. We need to be targeting the causes of ALS in what's called precision medicine. And the easiest ones, of course, to target are the genetic causes. And I think we've already seen the next home run gene therapy that's coming for a subset of ALS. This is an antisense oligonucleotide for FUS ALS, which is also, like SOD1, quite rare. So this was an article, a very small case series that was published from Columbia last year. And in that case series was an ALS reversal, somebody whose ALS was progressing, they had lost the ability to care for themselves. And after several months on this antisense oligonucleotide, all that came back. So it's tempting to think, why don't we just develop antisense oligonucleotides for all of the 40 different genetic subtypes of ALS? Unfortunately, they don't all cause ALS in the same way. So SOD1 mutations and FUS mutations, they kill motor neurons through something called the toxic gain of function. That means that the mutant protein is the problem. So the strategy is to get rid of as much of that as you can, and that's what you can do with antisense. Some of the genetic mutations, including the most common genetic abnormality to cause ALS, which is called C9orf72, are going to be more complicated than that. So we've already tried an antisense strategy for C9orf72, and it didn't work. And so we may need a cocktail approach, where there's something there to knock down the mutant protein, but something also to replace the wild type function. But more likely, we're going to get breakthroughs with this technology called gene editing. So instead of just grossly blocking the ability of a broken gene to make a broken protein, gene editing actually fixes the gene so that it can behave normally again. And it might sound like science fiction, but it's already happening. So there's a technology out there called CRISPR, and a lot of pediatric institutions are using this. When babies are born that have these inborn errors of metabolism, where they have what's called failure to thrive. They're not acting normal. They're not developing neurologically normally. They can't keep weight on. They can quickly figure out what's wrong with those babies genetically, and they can fix it with this gene editing technology. So we're curing a lot of diseases in kids now that used to kill them. The trick for using it for a disease like ALS is we don't know how to get it into the brain yet, but I think we're going to solve that. So I put a reference there about some people that are working on that very problem and have some really exciting ideas. And I would be very surprised if in the next 5 years, we don't figure out how to get CRISPR into the brain. And once we do, I think you're going to see home runs for all the genetic subsets of ALS. But what about everybody else? I said, only 15% of people have genetic ALS. Well, we have another technology that I'm very excited about. I told you before that we think sporadic ALS is complicated. It's probably combinations of susceptibility genes that open the door and environmental exposures that let the disease come in. And those combinations are not going to be the same. In other words, there isn't just one susceptibility gene and one exposure that's causing all sporadic ALS. We would have figured that out by now. There's probably dozens of subsets where each subset is defined by a handful of susceptibility genes and a handful of exposures. How in the world are we going to sort that out? Well, the good news is we've been building what I call big data for several years now. There's a number of large databases that have been asking patients with ALS to enroll. In the United States, we have the National ALS Registry, we have the ALS TDI ARC, we have the CREATE Consortium's database, We have the Answer ALS database. You know, altogether, these have tens of thousands of patients, and each patient has lots of information. They have information about their disease characteristics, about their concomitant medications, their comorbidities. They have information about their genetics, and they also now are starting to collect other omics, proteomics. Some are even collecting microbiome samples. So there's lots of data in there. And we've got this new tool called artificial intelligence. And if AI doesn't decide that we're not necessary anymore and wipe us out, I think it's actually going to solve a lot of these complex diseases. It's really good at finding patterns in large data. So again, I would be surprised if in the next 5 years we don't have a few subsets of sporadic ALS figured out in terms of exactly which susceptibility genes and which exposures are causing that subset. And that's the first step to getting home runs for those subsets. So my favorite subset, in case you don't know, are called outliers. I've always been fascinated by the people in my clinic who do extraordinarily well with ALS. And, you know, I'm not the first person to describe these folks. I mean, Stephen Hawking, you know, was diagnosed with ALS long before I was born. He was told he had 2 years to live, according to the book that was written about him and according to the movie I watched. And he lived more than 60 years with ALS and had an amazing life. This young lady in this picture is Andrea Pete. She's one of my patients. I'm not breaking any confidentiality. She has put me in her book and in her movie, and she's an extraordinary outlier. She's 12 years into her ALS and can still do marathons. Not exactly the same way she used to, but she does them in this recumbent trike. And she's actually the first person with ALS to do a marathon in every one of the 50 states with ALS. So incredible. So I'm fascinated by these patients. There's even a very extreme group of outliers that I call ALS reversals. And I define these as people whose records I have, where I'm confident that the diagnosis is correct, who've progressed to where they're disabled and then recovered all or most of their lost motor function. And there's 65 of them now that I've collected in a database. I know some people, I read that some people say this is, you know, Bedlack is making something up. These patients were described all the way back to the 1960s. All I'm doing is collecting them, coming up with a rigorous definition, collecting them and trying to study them. And so I've been studying outliers now for more than 15 years. And I'm happy to say I have found 3 things that I think could be possible explanations for them. And I'll tell you about those targets, and I'll tell you about how I'm trying to study them in clinical trials. So the first one is something called the gut microbiome. So inside all of us, in our mouths, our stomachs, our intestines, we have trillions of organisms. We have viruses, bacteria, and fungi. And it's been known for many years that alterations in the family of organisms there can cause diseases of the gut. Somebody got the Nobel Prize for discovering that peptic ulcers are caused by an overgrowth of one bacteria in the stomach microbiome. They can also cause a serious form of colitis called C. diff colitis. In recent years, people have started to wonder, is it possible that these organisms make things, chemicals that can get out of the gut and travel to the brain through something called the gut-brain axis? And I know I started to wonder about this when I saw 2 of my very slow progressing outliers get an infection, get antibiotics, and have explosive progression after that I could not get back under control. And I wondered, like, what happened? Did we mess up their gut microbiome? So we spent a few years collecting stool samples from patients in my clinic. And sure enough, there's a drastic difference in the composition of both the oral and the gut microbiome compared to people with typically fast progressing ALS, where the ALS FRSR slope is greater than 1.5 points per month, versus those with very slow progressing ALS, where the slope is less than 0.5 point per month. So the microbiomes of the fast progressing patients are way more complex, and those from the slow progressors are much simpler and very high in one bug, which is called P. vulgaris. That's interesting, but it's just an association. So luckily, I've got a lot of partners at Duke that have labs. One of the labs has the SOD1 mouse model. We took some of the stool from these patients from my clinic and we put it into the gut of that mouse. If we took stool from fast progressing patients, we could speed progression in these animals. If we then took stool from the slow progressing patients, or even if we just took isolated P. vulgaris from slow progressing patients. We could slow the progression right back down. So we're pretty excited by these. And naturally, we started to think about how could we manipulate the gut microbiome. So over the course of my life, usually when I get a good idea, I go to the literature and I see, damn, somebody already had this idea and they're already ahead of me on it. So sure enough, there are 3 published case reports from China that all suggest that fecal transplants might slow the progression of ALS. Now, those are just case reports. There's a lot of things going on in a case report. It's very difficult to know for sure if it was the fecal transplant that slowed the progression, but still interesting. Somebody actually did try a fecal transplant trial, but I don't think the way they did it was very scientific. And in fact, they admitted at the end that they didn't change the composition of the participant's gut microbiome. So it's not surprising that that trial showed no benefit in terms of progression on the ALS FRSR score. You don't change the gut microbiome, you're not going to change progression. So we have spent more than a year getting together with people who know a lot about changing the gut microbiome. Duke's got a fecal transplant clinic. There's a group at the University of Minnesota that has a product. It's actually purified stool from healthy donors, and they've got it encapsulated. The capsules are odorless and tasteless, and they've got trials going on with this capsule that the FDA has let them do. So we got all these different groups together, and we started a fecal transplant trial that's very different from the previous one. And we've got 8 patients enrolled. For the most part, the patients are too early in the trial to know what's going to happen. We're targeting a total of 20. But I did want to show you this because it's probably the most exciting thing I saw today. I cautioned you, it's only 1 patient. It's the first patient from our trial who just finished yesterday. And you can see her ALS FRSR progression over the period of time from October 2025, when she had her first score, to January 2026. She was losing about 2 points per month on the ALS functional rating scale. And then we cleaned out her microbiome. We gave her this fecal transplant. And as you can see, for the 6 months of the trial, there's really been no progression. Again, it's just 1 patient. It could just be a chance luck, but I'm encouraged nonetheless. And I hope we see more patients like this in the trial. So the second target that I found was exclusively found in those ALS reversals. So one of the things that I was able to do by partnering up with people who know a lot about genetics, like my buddy Michael Benatar at University of Miami and a group at the St. Jude, is do whole genome sequencing in these patients. And of course, when you're going to do that and you're going to look for new genes, you've got to come up with a way to look for controls. So we chose controls that were ethnicity matched because we didn't want to pick up genetic signals that were related to ethnicity. We wanted to focus on things related to possibly ALS progression. And these were patients with typically fast progressing ALS. And we found something. So about 1/3 of the reversals have a genetic abnormality that looks like it's in a promoter for a gene called IGF-BP7, insulin-like growth factor binding protein 7. And this specific single nucleotide variant has been associated with tissues that are in large biobanks. And what we can see is that this is associated with a lower expression of IGF-BP7 protein in brains and spinal cords of people who died from ALS. That's interesting. Well, no one's ever thought that IGF-BP7 might have anything to do with ALS, but there is a theory by which it could, and it harkens back to something called insulin-like growth factor 1, IGF-1. Once upon a time, I would say that IGF-1 was one of the most promising targets in ALS. People did studies and they found that the circulating levels of IGF-1 were related to how fast someone with ALS progressed. The more of this growth factor you had, the slower you progressed. They gave IGF-1 to the mouse model of ALS, dramatically slowed progression. And then they tried human trials and they gave people IGF-1 subcutaneously a couple times a day, and the trials didn't work. They saw no benefit. And so they scrapped the whole idea for a while until somebody came back around and said, maybe the idea is not wrong. Maybe the way you gave the IGF-1 was wrong. Like IGF-1 does not cross the blood-brain barrier. So giving it subcutaneously is not going to help a brain disease. And it also has a very short half-life. So giving it just a couple times a day, it's not going to be around long enough to help with neurodegeneration. So what if instead of giving people infusions of this growth factor into their spinal fluid, which would be very difficult, what if you could just knock down a protein that blocks the interaction between IGF-1 and motor neurons? Well, that's what IGF-BP7 does. One of its many functions is to block the interaction between that growth factor and motor neurons. So with less of it, the interaction can be stronger. Motor neurons can be more resistant to ALS. Well, since I published that back in 2024, there have now been 5 new observations, including one by a completely different group, that all support the idea that lowering IGF-BP7 is a reasonable therapeutic target. So we've already talked about ways to knock down proteins using gene therapies like antisense oligonucleotides. So I've got a couple labs at Duke working on that. It does take time to develop a gene therapy that can knock a protein down safely and get that into a human trial, but someday I hope to do it. In the meantime, as many of you know, I study supplements, and I have a big team that studies them with me. And one of my team members pointed out an obscure paper in the bodybuilding literature that talked about a supplement called hydroxy beta methylbutyrate, or HMB. It turns out that this is a performance-enhancing drug. It's actually banned in a lot of sports, but somebody did a trial where they gave some athletes who were training this supplement and others a placebo. And the ones who got this supplement had lower levels of many different biomarkers in their blood, including IGF-BP7. Fantastic, because supplements are something I can get into trials really quickly. And so I've got the protocol, I've got the FDA approval, I've got the IRB approval. I'm just trying to get a couple of contracts in place. And then hopefully in the next few months, we can launch this clinical trial of HMB, try to knock down IGF-BP7 and see if anybody else has an ALS reversal. So the last target I'll talk about is maybe the most surprising one of all, and that's hope. So I've talked to many, many slow progressors and almost all of the ALS reversals. And I can say the one thing that almost all of them share in common is they're very hopeful people. Now, I don't know if this could be the cause of why they're doing so well or an effect. I mean, if somebody sat you down and told you that you had 2 years to live and you were on year 12 and still doing marathons, you probably would be a lot more hopeful than you used to be. So it's hard to sort that out. But I will tell you that I think hope can be a treatment. So first, I think a lot of people have the wrong idea about what hope is. It is not just optimism. It's optimism plus agency. It's a state of positivity, but it's accompanied by having goals, finding pathways to achieve those goals, and finding motivations to go down those pathways. Hope is measurable. There's lots of different validated scales for measuring how much hope someone has. And if you look across many, many different diseases, people with more hope are doing better medically. So it's not just their quality of life, they're actually surviving longer. And you might say, how could that be? There's actually a biology to hope. So more hopeful people have different functional MRI brain activation patterns compared to less hopeful people. They also have different levels of chemicals circulating in their blood, many of which are involved in inflammation. So more hopeful people tend to have lower pro-inflammatory biomarkers in their blood compared to less hopeful people. So there's a biology. There also might be a practical reason. More hopeful people tend to be more compliant with evidence-based therapies. They're the ones who take the medicines like Riluzole. They're the ones who adhere to the exercises like respiratory muscle training and the diets like the high glycemic index diet. And less hopeful people don't. They don't see the point. And so it's not surprising that people who have more hope, who are more compliant with evidence-based treatments, might be doing better. And so I've asked myself, if hope is a treatment, where's it coming from? And I think for a lot of people, they bring it with them. It's either intrinsic, something they were born with, or it comes from what I call their often awesome army of family and friends, their social circle, their church group, their social media group. But a fair number of people look to their physicians for hope. Like, for example, when you survey people with very late stage cancer and ask, who do you look for in your life for hope? one of the most common answer is my doctor. And it turns out that in the cancer literature, there are papers identifying specific physician behaviors that boost hope, as well as behaviors that suppress hope. And they're not rocket science, but that doesn't mean they won't be easy. They're going to be challenged because, first of all, doctors don't get any education on hope. Doctors are under tremendous time pressure, and some of these behaviors take time. And unfortunately, many doctors, especially in my country, are burned out. And what is burnout? It means you've lost your empathy. That's probably the most important thing that you need to boost hope is empathy. So I've developed this whole approach, multi-year approach to boosting hope. I call it Stitching Strength. I've written an editorial about it, which you can find at that reference. I've got a movie about it. You can see a preview for the movie by scanning that QR code. I have an Instagram account where I explore different hope boosting strategies, including different outfits and silly things to try to make people laugh and boost their hope. But my approach involves the following. First, self-care. Boosting hope has to start with me. Like if I'm miserable when I go to clinic, I can't be a hope booster. I have to understand what my body needs as far as sleep and nutrition and, exercise. I also have to figure out what things bring me joy and incorporate as many of those into my life as I can. And for me, ever since I was a kid, it's been the clothes. Like I need the bright colors. They just lift me. They're like a suit of armor and they keep me resilient to any kind of stress I might run into. But it's more than that. When I get into the room, I have to be careful about the words that I choose. I don't like to use expiration dates. I've never understood why when somebody is diagnosing a patient with ALS, they tell them that you have 2 to 3 years. How do you know, looking at that person, that they have 2 to 3 years? I mean, Stephen Hawking went for 20. There's many of my patients who've lived for more than 10 years. So you really can't look at a person the first day you meet them and know they're going to live 2 to 3 years. I like to focus on what we can do. And there's a lot we can do, as I've already talked about. I use if things get worse when we talk about progression. If things get worse, next time we might have to talk about this difficult option, not when things get worse, because I don't know for sure that things are going to be worse next time. When we have to have difficult conversations about things like a feeding tube or a ventilator or goals of care, I talk about them as a plan A and a plan B. Look, plan A, we'll try something new. If you want to keep experimenting, I'll keep finding things for you. And I hope that works. I hope that's the one that slows, stops, or reverses your ALS. But if it doesn't, I need your help with a plan B. Like, we don't want to just pretend that this couldn't get worse. We should be prepared for any circumstance. Part of hope boosting is getting to know each person. Like, this is not ALS sitting across from me in the clinic room. It's a person. ALS is part of their story, but they're much more than that. And so I need to understand the things that bring them joy, what their support system looks like, what specifically they're hopeful about when I see them in clinic. And I've had patients, where the measurements that we make are worse than they were the time before. And I see how it weighs on them. And in fact, I remember reading a blog of a professor who had ALS at the University of Virginia. And he got home from his clinic and he said, every measurement they made on me is worse. And I'm so depressed. I tried so hard. And he came up with something. He said, what if the clinics measured something else that didn't have to be worse? And he invented it. It's called the turbocharged living tool. And I would encourage you to take a look at that because it asks people about their kindness, their compassion, their humor, much more important parts of a person than motor neurons. And I think it's a great complement to the other things that we measure. So I know I'm running short on time. I don't think everyone's going to be able to boost hope the way I do. It's just incredibly time consuming. Like, this is my whole life. And I think some people want a work-life balance. So I'm working on a specific hope booster. And in short, it's basically asking people what they're hopeful about and using the turbocharged living tool. And I'll tell you, the early experience with this looks very promising. So we have a very nice trend emerging in our first 40 patients where over 2 visits, those who got the intervention, their hope scores appear to be shifting to the left, meaning increasing, whereas people that didn't get it, they're equally likely to be the same or worse or better. Also, we see that hope in ALS patients is definitely correlated with satisfaction with life. So in the last few minutes, I'll just run through these slides quickly. I wanted to make sure that people knew there was something out there for everyone. I know sometimes people get upset that they can't find things to participate in, There's natural history studies all over the world. Like in the United States, we have many. I'm sure they have many in Europe. You probably have some in New Zealand and Australia. These are so important. They're not treatment trials, but this is where you contribute to data that AI will use to help us understand why this is happening and why some people are doing so much better. Obviously, most people want to be in a clinical trial, and there's more today than there's ever been, still not nearly enough. And I know they're often restrictive and difficult. Expanded access programs are starting to happen. Here in the US, we've had a lot more in the past 5 years because of something called the Act for ALS that President Biden signed into law 5 years ago. We're hoping that gets reauthorized, but still across the world, I know there's not nearly enough. And so many of you know I run a program called ALS Untangled. We scientifically review any therapy, any unusual therapy that's out there. I call these alternative and off-label treatments, and we have found some that we think are pretty promising. So for people who can't find a trial or an expanded access program, I tell them these are the most promising things I know of today to self-experiment with: acetyl L-carnitine, antiretrovirals, clenbuterol, L-serine, methylcobalamin, nicotinamide riboside, and pterostilbene, quinidine and dextromethorphan, and tamoxifen. I would just say be careful out there. I understand why people go to the internet in this day and age to try to find their hope, especially if they're not getting it from their own doctor. But there are many people on the internet who are selling false hope. False hope means that they've distorted reality. Somebody who tells you if you give them $70,000, they guarantee they can reverse your ALS, that's false hope. There's no data to support a claim like that. So just to summarize what I told you about today, I think it's an incredibly exciting time. I've been doing it for a while, and I've never been more excited to go to work than I am today. We've got all these new clinical options that slow progression. We've got options now for the first time that improve motor function. I think we're on the cusp of understanding more causes of ALS, and that's going to mean more home runs. I'm starting to understand, I think, why some people are doing so much better, and I think I'll soon be able to make that happen more often. And in the meantime, there's lots of opportunities for people living with ALS who want to get involved in research or try experimental products. And so I'm sorry I went so long, but I will stop here and I will try to take your questions in the last few minutes.
Thanks so much, Dr. Bedlack. That's just a fantastic presentation. We do have a small range of questions, which are from very personal, specific questions to a couple of very scientific ones. James, can I leave you to perhaps ask the first question on behalf of our audience?
Thanks, Mark. So this one's actually, I'm going to start with a scientific question, probably, well, somewhere between scientific and clinical. Somebody's asked about thoughts about a keto carnivore diet.
Yeah. A keto diet, did you say?
Yeah, a keto or a carnivore, what's been termed a carnivore diet. I guess they are different things, but there are relationships.
Yeah, so we did publish an ALS Untangled about this. So like a lot of these things, there is a theory by which this could help. So when you eat a diet that puts you into ketosis, your body starts to make things called ketone bodies, and they can be used as a source of energy by cells who are having trouble making energy the old-fashioned way through the mitochondria, which is almost certainly happening in many people with ALS. So there's a theory, but that's kind of it. Like there was an attempted trial of a ketogenic diet. It closed without an answer because it enrolled so poorly. We actually did a trial of something different than a ketogenic diet. It was actually sort of a super powerful form of coconut oil, a medium chain triglyceride that would boost ketone bodies much more profoundly than a ketogenic diet. It didn't do anything. It didn't slow ALS progression in our small trial. And the thing I worry most about is that ketogenic diets cause weight loss. And so when I put up, the pros and the cons, the pros are there's a theory, but there's really no proof of it. The cons are, I know it's going to cause weight loss, and I know weight loss is bad for people with ALS. So to me, that's not a great option. I think it's more likely to cause harm than to help.
Great, thank you. A related question, this one about the high glycemic index diet, a question about timing of meals. Is it better to eat smaller, more frequent meals through the day or 3 standard meals on the high glycemic diet?
Yeah, I wish I knew the answer, James. These are the kinds of questions that you could answer if you did a trial. But when it's a cohort study, I mean, you only get back what you ask. So I don't believe they asked people like specifically how many meals a day. I think they were asking people the kind of things they ate, and then they were using those food diaries to classify people's diets. But I don't think they asked that question. And yeah, there's a million questions, to ask out of this. I mean, we already talked about the fact that ALS might not be the same disease. Is it possible that this high glycemic index diet is better for some subset of ALS? Again, I think you can only answer questions like that with a clinical trial, and those are hard to do when it comes to diets.
Thanks. We have a question from Dr. Emma Scotter, who leads the MND team at the Spectacular Centre for Brain Research here in Auckland, New Zealand. And Emma asks, how do you reconcile the fecal transplants / diverse gut microbiome protective effect with the protective effect of a high glycemic index, which I believe reduces microbiome diversity?
Yeah, so I actually say if the high glycemic index diet reduces microbial diversity, that's probably a good thing. So we talked about the fact that comparing the microbiome of fast and slow progressing patients, the fast progressing patients are the one who have the most complex, diverse microbiome. The slow progressing ones have the simplest gut microbiome that's really high in P. vulgaris. So I don't know of any studies that look at that high glycemic index diet and how it affects the gut microbiome. But if that's true, that's really fascinating because I think it supports what we found in our patients from our clinic.
Thank you.
There's a clinical question here that is an interesting one. So this particular person has ALS and diagnosed earlier this year with abdominal pain that feels like a stitch, sort of like a diaphragmatic cramp, I guess, after doing a long run. In your experience, have you encountered this? Is this muscle pain? Does this abdominal pain sound like it could be related to the motor neuron disease?
Yeah, it's a great question, James. I would have to know more about it. I mean, if it's something that comes and goes, then yeah, I mean, you can definitely get severe cramps in your abdominal muscles. You can even get, cramps in your diaphragm and your larynx. These can be extraordinarily uncomfortable, but they come and go. So, I mean, a person who has just persistent severe abdominal pain, I would have a hard time understanding how that could be related to ALS. I would want to look for other causes there.
We're about a minute left. A question about ozone therapy, whether there's a suitable alternative treatment option.
Yeah, that's another one that we did an ALS Untangled review on. And I would say there's a theory that has to do with oxidative stress and neuroinflammation, but there really isn't any data to support the use of it. And in fact, the way that many clinics are doing it is quite dangerous. So the FDA has even come out and warned people against doing inhaled ozone therapy because it can cause bronchospasm and death. And it's kind of amazing. Like there's a lot of things happening out there that are hard to believe. I mean, I remember when I was very interested in curcumin, and I spent a few years doing a clinical trial. At one time, that was my #1 alternative and off-label treatment. It doesn't work. I tried it. It doesn't work. But I mean, there were clinics. I came across clinics that were actually injecting it. And there were associations with death, with sudden death from injected curcumin. So it's it's amazing what's going on out there. I would just be very careful. Use those ALS Untangled 10 red flags. Don't hesitate to reach out. If you can't, navigate the ALS Untangled page or your doctor won't help you, just reach out to me. I'll tell you if we've looked at it, if I have any data on it, and what I know about it.
Thank you. I just see we're up to 1 o'clock now. We've pretty much run out of time. We still have a range of questions, and look, we'll undertake to collate those questions and pass them on where it makes sense to do so. If you're up for that, Dr. Bedlack, it sounds like you have an interest in that area, and there are quite a few other varied questions here again. And I'd just like to say on behalf of all of our participants today, we've had about 200 people attending throughout the presentation, and from this community to yours, we'd just say thank you so much for your attendance, your delivery. The richness of your commentary today has been fantastic, and I know that people here in New Zealand are really, really appreciating that. I'll hand over to you, James.
Thanks, Mark. I would absolutely echo that. Look, I found this talk absolutely fascinating, and look, I think you've reinvigorated hope in many of the people who who look after patients with MND, and it's just fantastic to see someone who's such a a leader and such passion for MND, and to be able to hear you speak is fantastic, Rick. Thanks so much.
I appreciate the invitation, and I especially appreciate people living with ALS who are out there on the call. I know it's not always easy for you to make these kinds of things, and I really appreciate, your support. It's part of the reason I'm still here after 25 years. There's days when I feel like giving up, and it seems like every time I'm near quitting, I get a message from one of you, or I see something on social media that one of you are doing, and it just brings me back and tells me we got to stay in this fight together. So.
Absolutely. Thank you again. And there are a lot of thanks in the questions and answers as well. We'll pass those on as well, Dr. Bedlack. And thank you so much for your time. We'll wind up that now. And as mentioned, the video recording will be made available to every registrant as well. Thank you all. Have a good afternoon and have a good evening, Dr. Bedlack.
All right. Take care.